Hello, I'm Rhalena Thomas. I'm going to talk to you today about my work with Dr. Ted Fawn on midbrain organoids as a model for Parkinson's disease, a disease that is marked by loss of control of the motor system. The main area in the brain affected in Parkinson's disease is the substantia nigra within the midbrain. Dopaminergic neurons create a black color and can be seen in the midbrain. In patients with Parkinson's disease, these dopaminergic neurons are subsequently lost. Currently, there is no cure for Parkinson's disease, and although some genetic and environmental links have been identified, the causative pathways remain unclear. Immortalized cell lines in animal models are not sufficient to study Parkinson's disease. In the Fawn lab, we can take skin or blood cells, reprogram these into induced pluripotent stem cells, which can be differentiated into different cell types, including neurons, or they can be grown as a 3D tissue, which we refer to as human midbrain organoids. To understand the cell types within these human midbrain organoids, I performed single-cell sequencing analysis, in which organoids are dissociated into a single-cell solution and then processed using the 10x chromium genomics system, where a bead containing all of the chemicals needed for RNA amplification and a cell are merged together. These are then sequenced, and each cell's transcriptome is found. Further, I analyzed these using an unbiased machine learning to create clusters or groups of cells, where the cells within one group have the most similar expression to each other and the most different expression from all of the other groups. To identify the cell types within the groups, I took the top most differentially expressed genes between groups, as well as candidate markers that are known to be distinctive between cell types. The examples I'm showing here are SOX2, a marker of pluripotency, MAP2, a marker of neurons, S100 beta, a marker of astrocytes, and oligo2, which can be a marker of oligodendrocyte precursors. I then annotated the cells, and I found that I have two clusters of neurons, a cluster of dopaminergic neurons together with neural precursor cells, oligodendrocyte precursors, astrocytes, and radioglia, which can become neurons or glia. There was also a group of mixed cells containing all of these cell types, as well as other cells. Single-cell sequencing is a powerful tool, but also very expensive, and not necessary to just distinguish large groups of cells within the organoids, and it can't be used to process a lot of different genotypes or replicates. To address this, I've been developing the use of flow cytometry to quantify cell types within organoids. For this, the cells are still dissociated into a single-cell suspension, and then they're labeled with a panel of antibodies. And then flow cytometry measurements tells us the intensity of staining on each individual cell. The data has to be processed to identify the single cells that are alive, and then the intensity of each antibody can be measured. I again used unsupervised clustering to create different cell groups. For this data, I created a matrix of the predicted expression for each of the antibodies that I've used, indicated on the y-axis, for the different cell types indicated on the x-axis, which should be within the organoid. I then labeled these cell types, and I found the same cells as in the single-cell sequencing, and now I also identify stem cells as well as epithelial cells. To understand the disease pathology, I'm using two models of Parkinson's disease that cause a genetic form of Parkinson's disease that is early onset. PINK1 and Parkin are those genes. We've deleted each of those genes in a control cell line and created midbrain organoids from these genotypes. I performed bulk RNA sequencing, where the whole organoids were homogenized and RNA was extracted and then sequenced, aligned to the genome, and then I calculated the differential expression between each of these genes to look for up and down regulated genes. I performed ontology analysis, and I find that there are common pathways in PINK1 and Parkin deletions that are up and down regulated. The pathways where the genes were upregulated were all related to synaptic transmission. and the pathways that were downregulated were all regulated to the extracellular matrix and cytoskeleton. To determine if this represents a change in the cell population within the organoids or changes in a small percentage of cells, I'm going to perform flow cytometry and single cell sequencing on these same organoids. Thank you for your time. If you have any questions, I can be contacted on the information shown on the slide.